Um, hello, welcome everybody to this discussion session for the CBPF Summer School. I am Eugenio Piasini, and we are here with the other. Let me just start with sharing the screen. Okay. Okay, that is better. Here, this is discussion sessions for today's talk by Professor Balasubramanian, How the Brain Interprets Complex Signals. We had 3 papers that were assigned reading for today. And so I'm Eugenio Piasini. I'm a postdoctoral fellow of the Computational Neuroscience Initiative at Penn. And the other, we have the other speaker today. Ron, do you want to introduce yourself?
Sure. Hi, I'm Ron DiTullio. I'm a graduate student working with Vijay Balasubramanian, as well as Professor Yale Cohen at UPenn.
Cool. As we can see today, we had 3 papers. First one is the by Eli 2015 about grid cells. Second one is a Neurocomputation 1997 paper about Occam's razor. And the third one is the Ratliff PNAS 2010 about retina. So we're gonna go over them in order, looking at the questions you've sent, and we'll try to address them as best as we can. If during the session you want to ask more questions, you can type them in the Q&A panel, and we're going to try to, I guess, address them by the end of the in between papers or at the end. And if you have more questions, you can see our emails up on the screen. You can always get in touch with us. Yeah, and that's it. If we don't get to to answer everything by the end of the session, we'll try to put up the answer somewhere. Cool. Ron, do you want to get started with the grid cell paper?
Certainly.
Stop sharing so that you can
pull up my slides. I'm going to keep the questions on the side. So I will now start sharing. Go. See this, and then hopefully I'll hit present. So hopefully everyone can see the system. This is great. Yeah, so by what means of a slight introduction, or a thing I always like to show when we're discussing grid cells, is actually the complexity of the full circuit. And this will then tie into kind of some of the questions that were asked. The main thing, so what we're looking at right now is current as of 2015. I think there's actually a more updated version of this that came out recently. But is the actual full functional circuit that is associated with grid cells and place cells, so the entorhinal cortex, areas before the entorhinal cortex as well as the hippocampus. And the reason why, again, I bring up this image is there's actually a ton going on in this circuit, and the paper that we're looking at, this grid cell paper, is really focusing on the medial entorhinal cortex, just one box in this vast array of different information. So I'm gonna, as I answer questions or as people come up with more questions, I'm gonna give you, the best answers that I can. But I think, yeah, there's just a lot of open questions that are still left in the field. So sometimes we won't have the most satisfying answer, but maybe we'll have inspiration for new experiments and new work going forward. And I think the first question that we ended up getting asked was if grid cells respond the same way when an animal is in a similar environment, or does this only occur when an animal is in a novel environment? There are differences between how grid cells respond in novel versus similar environments, but overall that hexagonal pattern that you see and the periodicity and the modules that the paper was modeling is seen regardless if the environment is new to the animal. So there is a bit of a lapse on this. So the main reason for that is that to see that a cell is a grid cell, we need the animal to have explored a certain amount of the environment. And obviously our animals can only move so fast. So one open question still is what counts as novel to the animal? Like how quickly does it actually learn its surroundings or learn the environment? But overall, I would say you will see a grid pattern regardless of the novelty of the environments of the animal, just sometimes certain properties of the grid pattern are changed. So for example, there's a 2000 paper or 2007 paper from Caswell Berry's lab showing that the grid cell, the distance between the fields seems to expand or be broader when the animal is in a novel environment. But overall, you will always see the grid pattern even if the environment is novel to you. The second part, I think, of that question was asking then, okay, if these grid cells exist in the dark, though, what is the exact input of visual, or what is the impact of visual input onto the circuit? And that's an excellent question. That's another reason why I brought this graphic, is so these areas that are feeding into the neuronal cortex, and to orient on this graphic a little bit. So this LEC is the lateral entorhinal cortex, the MEC is the medial entorhinal cortex, so two sections of the same brain region. And the medial entorhinal cortex is the one where we find grid cells. But obviously, since these are two parts of the same brain region, they have a lot of crosstalk, observed by these arrows. And the lateral entorhinal cortex actually receives a lot of visual input, as does the MEC, but the LEC in particular receives a lot of visual input. So the interaction, or the way that visual input gets into the neuronal cortex, is probably through these LEC connections, combined with some representations of, like, for example, environmental boundaries. So there are cells located in these regions, the subiculum, that will become active when an animal is really close to a wall. And those cells will also kind of, or in a way, are providing visual input, the visual input that you are near a boundary to the medial cortex. So the short answer for how does visual input kind of play a role in it is quite complicated. But essentially, the inputting regions to the neuronal cortex, both the lateral neuronal cortex and previous sensory regions, seems to provide some visual input, and how exactly that influences grid cell firing properties is still under investigation, both through changing just the visual inputs of environments, and more recently, we can have rats in particular run on trackballs, and you can create a virtual environment around them. And you can try, like Lisa Giacomo in her lab has an excellent experiment where they modulate the rate at which the visual world goes by, kind of counter to how the rat is running. So usually when you're running at a set speed, you know, things go by you at a certain speed, versus they mess with the optical flow intentionally in this paradigm, and they do see some changes to the grid pattern caused by that. So we're still investigating exactly how visual inputs affect grid cells, but they definitely do, and there's definitely regions that are providing really high level visual input into the circuit. I think I see something. Okay. Yeah, so I think that answers question one. I'm just going to real quickly look at the other question that we have. I just need to get out of presenter mode, that's right. So that was question one. Oh yeah, it's logarithm theory. Yeah, so for the behavioral range, not sure, sorry, missed my click there. For the behavioral range of the modules, that's another excellent question. It's another thing that's actually under active exploration. So particularly, most of the work in grid cells has been done in rodents, typically mice or rats. And for the scale of grid cells and even place cells, we've been really limited just by the physical size of the environments that we generate. So when we're recording from an animal, usually most of the traditional papers, you're seeing environments that are maybe 1 meter square to 1.5 meters square, maximally maybe 2 meters square. And it's only recently that actually someone I mentioned earlier, Caswell Barry, who's at, if I'm not mistaken, the University College of London, he's now looking into much, much larger environments and the influence that has actually on the grid cell pattern and the ability to see these larger and larger fields. So the actual relationship between these different grid cells and the natural environmental boundaries that this particular rodent or rat will run into is still something we're actively exploring. But the theory does allow, really predicts the scaling in between modules. And that shouldn't change drastically even if you are increasing the range. Like the actual ratios between different modules should scale at the same approximate rate, and the theory does align with that. And so far from the more recent results we've gotten, or that we've heard about, at least through Caswell Barry and others, it seems that the theory holds. But that's definitely something, as we get these recordings of larger and larger environments, we'll have a better idea of how correct this is in these larger contexts and potentially more realistic contexts. All right. And then should put the questions in the slides. Third one, what techniques can we use for grid cell visualization? So this is a great question. So I'm actually going to skip this slide. So yeah, so mostly grid cell visualization, if you're talking about just how do we plot the data, is usually presented in these kind of two very basic ways. So in this case, what are we looking at? So on the left here, this black line represents wherever a rat was running around inside the square box. I think this one's about 1.25 meters by 1.25 meters. And these red dots signify whenever a grid cell was active, whenever that cell fired an action potential. So you can usually see, I mean, what may be striking at first is even just looking at this raw image where we're just seeing where spikes were fired over the animal's trajectory, you can really see the grid pattern very obviously. Often a lot of papers, you more often see visualization as we see on the right, where this is taking the same image, but normalizing by occupancy, by the amount of time an animal has spent in each position, and then scaling things accordingly so that, you know, the dark red colors indicate that a lot of spikes were fired, dark blue indicates that no spikes were fired. And typically when you visualize grid cells, you will see these plots. The only third one that I'm not showing here that's sometimes used is to determine the geometric properties of these grid cells. So example, like how far are two fields apart. Typically we'll take this occupancy normalized rate map, and then we'll do the spatial autocorrelation of it. And using that spatial autocorrelation, we'll perform statistics essentially on that image to try and determine either the orientation or the distance between fields or other properties of the grid cell. In case the question was, how do we visualize them actually in the brain? So the ways that they're recorded, most grid cells have been recorded using electrical probes. So we will actually put electrodes into the brains of, and typically rodents, and try to record the electrical signals of the cells we find there. An interesting thing that's been done more recently is you can also, in mice, manipulate their genetics so that you can cause cells to biofluoresce when they're active. So recently, there was a really interesting paper doing that in the entorhinal cortex that actually confirmed or helped confirm the current theory of how grid cells are generated, potentially, or help provide experimental evidence, at least for that theory. In that case, we're doing what we call calcium imaging. So cells, when they become active, they have an influx of calcium. And if you have this genetic manipulation, this influx of calcium will cause fluorescence. So then we can actually image a lot of cells being active at the same time. Yeah, I think that mostly addresses grid cell visualization. If people have more questions, again, feel free to email me or chat. Yeah, and then how broadly can the model be applied to different species? So I'll come back to this image. So that's a great question. The majority, again, of the work in grid cells has been done in rodents. There has been some work in bats. That's really exciting. Recently, I think there's started to be some work in birds, and there's very little in primates and humans. But one really nice thing, and I think we'll talk about this more again later as well, is that the structure where we find these spatial signals, that is the interauditory cortex and the hippocampus, tends to be well conserved. So what we have here in this paper, they go into more detail as well, but we can kind of visually see on the top, this is a cross-section of the human hippocampus and interauditory cortex, and on the bottom is the rodent hippocampus. And the very convenient thing is that they essentially, although there is kind of a weird rotation has occurred, they are remarkably similar. And even the subregions and the connections between the subregions tend to be remarkably similar between humans and rodents. Even to the point where the actual naming of these brain regions, so we see here on the right is a cross-section cut of the hippocampus, showing the unique subregions that are a part of it. We can maintain the same name of CA1, CA2, CA3. It's similar to the actual rodent. So there's a great amount of homology between rodents and humans in this region, and generally this region is well conserved. So we're optimistic that the principles that we're kind of finding in rodents would then generalize to other, mammals. On that same note, though, with bats, we've already seen that the grid cells in bats and the ways that they are organized is different, which kind of makes sense. It would have been very surprising if they had been the same, mainly because obviously bats can fly around versus we are kind of stuck to move in 2D. If you make a bat crawl on the ground, which you can argue is not super realistic, you can get grid cells that look kind of similar, but the mechanism of generation of these grid cells seems to be different. So if they're organized in these modules, that would also most likely be different. But I guess the good news is for trying to expand this to rodents or from rodents to humans and primates, that seems like that should be possible. There might be differences there, but we have more reason to be encouraged by that. But for other animals, especially if the navigation challenge is very different like bats, then we don't anticipate this theory to translate to them. We don't anticipate this to apply exactly in the same way. And I think those are the 4 questions for the grid cell paper. So I'm going to stop sharing my screen and then look at the question answer, see if anything else.
Yeah, I think we have a more of a so look, we have a couple of questions. I'll let you address the other ones. But so first of all, let me just say regarding the first one, we had a question about study materials. Ron, I think we don't have any problems for us. We can share our presentations with the
oh yeah, of course.
students in our school. I don't know what other study materials you're thinking of, and I'll let you take over the other ones.
Yeah, all these studies done in organoids. Oh, that's an interesting question. So one big issue with grid cells is there's not a single cell type, a morphological cell type that is always a grid cell. And grid cells are actually decently rare in the brain, so the relative sparsity in the rodent brain is pretty significant. So because of that, and because of the fact to tell a grid cell is a grid cell, you need the animal to be running around so I can record spikes from it as it moves. We can't really do or easily bring studies over to organoids. Now, it's not completely a lost cause because there are 2 types of cells that have morphological properties that we can identify. So the pyramidal cells and stellate cells of the entorhinal cortex. And those cells we can do slice physiology on. So often we will take a rodent that we sacrifice, we'll take slices of the brain, and then we can find the electrophysiological properties of these stellate cells and pyramidal cells and try to relate that back to what we know about grid cells. So for example, one thing that's very relevant to the paper we read with this is, they're saying, well, the grid cell scale changes over the length of the dorsoventral axis. And through this slice physiology, we do have an idea of how that might actually happen. And it's specifically that there's a certain channel type known as the H channel, or the H current channel, HCN, that seems to change its concentration in a systematic way as well along the dorsoventral axis. This channel has relationships to how a cell will respond to inhibition, to inhibitory signals being sent to the cell. And we have also done studies where if you do knock out this cell type in mice, you kind of reduce the dorsoventral gradient. So no, it's not done in organoids. We do some slice physiology of rodent brains. We have gotten some good insights from that. But the primary difficulty is that grid cells are really a functional cell type, which I only know that that's a grid cell by having the animal run around. And we don't yet have a biomarker or anything of that nature that we can use to be confident that we're definitely recording from a grid cell and slice. So when the animal's not, alive and moving. Yeah, so next question.
Yeah, Ron, if you want, maybe we can, if you want to,
what do you want?
if you want to, if you say one that you want to answer right now, then otherwise maybe we can just, maybe if you want, otherwise we can just keep moving and then keep, I just don't want this to,
that's fair.
Too much space, but then we can, I don't know, if you want to do another one now, and then we can see at the end, sampling a few of these, otherwise we can move on as you wish.
Yeah, I'll do one more, and then these are good questions, so I'm happy to come back to them. And then, as Eugenio said at the outset, feel free to email us. We're always, I would say we're always happy to chat. I'll speak for both of us. I'll take that arrogance. But okay, so let's see. So you have 3D. So in this, I guess I'll start with a question from Krishna. So they're asking, could we induce ambiguity in the internalization through manipulating the environment? So that is, that's a good question. So there is interactions between the environmental boundaries in particular and grid cells. So I know some work that I was recently working on is actually those border cells that I think I mentioned briefly before when talking about how visual inputs affect grid cells. We do have reasons to theorize that border cells interact with grid cells and actually will influence their firing properties. So yes, by like, for example, stretching the environment or doing other different things like that, that can cause changes in the grid cell firing pattern, at least once the change has initially been done. So if I have an animal running around a box, and then I take him out of the box and stretch the environment, put him right back in, then that will cause changes in the grid cell firing pattern. More than that is there can be contextual influences, though this usually isn't on grid cells. You can see this on another type of cells called place cells. So there's some really cool papers where even just by changing the light conditions in a room, where an animal is really used to a certain room always being in dark or a certain room always being light. If you are in the dark room and you flip the lights, you can see actually patterns in the brain where the animal's trying to figure out which context is it in again, or you can create ambiguity through that. But definitely there is an interaction in the system between the context and kind of your internal representation of where you are in that context or what map you're kind of loading or thinking of. I think I'll end on that question because that's a fun and inspiring. Throw it over to you, Eugenio.
Okay. Cool. Very, very nice. Thanks, Ron. Then, as I said, maybe at the end, we see what, if we have time at the end, we see what questions we have, and then we can come back to some of those. Then I'll share my screen, and we can move on to the second paper, which is the Occam razor paper. So regarding this paper, there was a little bit of general confusion. Looking at the questions, there was a little bit of general confusion regarding the kind of general math that goes into this. So I'm just gonna give a very short overview of how Bayesian model selection, of what Bayesian model selection tells us about how we may go about measuring the complexity of a statistical model, so that hopefully everybody is a bit more up to speed with this. So first of all, because there were a few questions, when we say Bayesian, we mean like we're thinking about general settings of Bayesian statistics, which is that settings where you think of probabilities as of degrees of belief in something, say in a state of the world or something, right? This is sometimes opposed to things such as frequency statistics, where probabilities are infinite limits of relative frequencies. Thinking about flipping a coin many, many times, the probabilities, is the limit of fractional time it comes up head rather than tails as you take infinite flips. But so it's important that here we're talking about Bayesian statistics because this forces us to make explicit our probabilistic assumptions about the world before we actually go out and make experiments in a way that I'll clarify in a second. So here's the general context. Say that you have some empirical data E, okay, E is some data set, right? It is something that you observe, you measure, right? Then the probable model selection is as follows. For simplicity, imagine that you have two models, right? This is something that you decide because this is your problem. You set it up as you want. So say that you have two models, M1 and M2. There are possible models for this data. Then because you're in a Bayesian setting, you have to also specify upfront how confident you are, what your degree of belief, is in each of those two models before you actually go and see the data. So this is what we call the prior over the model. So in this context, for simplicity, we're gonna, imagine that this is 50-50. You're observing some noisy data. You have two possible explanations for it, and a priori, before observing the data, you don't really know which one it is. You say, oh, it's could be either of those with equal probability. And then because each model is actually a parametric model, so a model in this context, as you've seen from the paper, is meant as a parametric family of probability distributions. So these models will have parameters so that we can summarize as theta. And so we also have to specify a prior distribution over the parameters of the model for each model. So for instance, if one model is a Gaussian with, say, a set standard deviation and an unknown mean, you need to specify a prior distribution over this unknown mean. So this would be your theta parameter, right? So this is the initial setting of the problem, and the problem of model selection is how do I go from these a priori assumptions on what my possible models are, and how do I combine them with the empirical data I observe to yield what we call a posterior distributions over models. So the degree of belief that I assign to each of my two models after observing the data. Right? So then all the above, just by the, just essentially by construction, implies the following. So the fact of specifying a model in, more concretely, means that we're giving it, essentially we have a likelihood function for the data E under model N. So the likelihood function is what we, sometimes we think of it as the model. So it's the function that tells, if it's a Gaussian, it's the function that, that with the, with the exponential and the square of the difference from the mean and so on and so forth. So this is, this is part of specifying the model. Then from just these definitions, we can define a couple of useful mathematical objects. So one is the evidence. So the evidence, or marginal likelihood, is this object that you see here. So this is the probability of observing the data given a model after you have integrated away the dependency of the parameters. Because of course, if you see just here above, the likelihood function tells you, if I have a particular model and if I have a particular choice of the parameters for that model, then what is the probability of observing the data that I have? The evidence, by contrast, integrates away the dependence on the parameters, as you can see here. And to integrate it away, of course, you have to provide a probability distribution over these parameters, and this is where our prior comes in. Oh, by the way, I'm gonna say something a little bit more about this prior that I've written here. Assume uninformative. This may be a little bit unclear for some of you. There is another question on this, so we're gonna look at it in a couple of slides. But the idea is that this object, which is the probability distribution, you can think about it as probability distribution of states of the world E, given that a certain model is true, okay? And this is an interesting object that we care about because of what follows. So in the end, for solving the model selection problem, what we care about is this posterior probability over model 1 and model 2. This is what I was talking about earlier, right? So you want to compute the probability of, say, model 1 after you have observed your data. So this is really by construction, this is the definition, sorry. This is the solution to the model selection problem, right? You want to attribute the probability of model 1 versus model 2. But just by application of the rules of probability, this is Bayes' theorem, you can write it as follows. You can just write it as a product of the ratio between the posteriors over the two models can be written as the product of the ratio of the two evidences for the two models multiplied by the ratio of the priors. But if because of simplicity, as I was saying earlier, we assume a uniform prior over the model, then this term cancels, and essentially what we have is that our posterior over models, which is what we care about for solving the model selection, is really given by the evidence. So computing the evidence is what we have to do. So and in the next slide we're going to see how exactly we go about this. But for the moment, just to give you an intuitive picture, where is the link between complexity and model selection? So remember that this complexity is, so this is a probability distribution, right? It's a proper normalized probability distribution. So, given a particular model, you have a certain distribution of the states of the world. And you can think about it as, this way. So if you have the two models, the red model and the green model, say M1 and M2, they may look like this, where this x-axis here is an abstract axis containing all the possible states of the world, and this gives you some probability distribution, right? So, and the idea here is that we identify, so, maybe these three distributions are such that one of them is broader, right? It's more spread out, and it kind of covers, a larger range of possible states of the world. So this model can possibly, by appropriate setting of its parameters, can explain decently well a larger number of states of the world compared to the green model, which has a narrower evidence. So a narrower marginal likelihood. So even if you move around its parameters, you can only explain well a smaller subset of states of the world. So we essentially identify then the model which is more spread out as the more complex, because it kind of contains more explanations, right? More different explanations. And so just by, this is just a very super hand-wavy explanation. By looking at the figure here, you can tell that, if I happen to observe, if my data E is something that happens to be explained by both of the models, so it's something that falls within the range of explainable states for both models, then because the red model is more spread out, the probability that it will assign to each of the states that it explains, it's also going to be lower. Whereas the narrower one, it's also will tend to be higher up. So we'll tend to assign a higher probability here. So from this diagram, maybe, hopefully you can have an intuition about the fact that models that explain narrower sets of states of the world tend to assign higher marginal likelihood to them compared to models that are broader. So this is kind of the general idea. So this is in terms of model selection and Bayesian statistics. Now we can move on to we said that we care about the evidence, right? So the other key passage is then computing this evidence. In general, this is some complicated integral that we don't know how to do. But one of the key points of the paper is that when your data set is large, where there are a number of data points and under appropriate regularity assumptions, when you have a large number of data points, you can write this integral as with a nice approximation that breaks out in multiple different terms that have pleasant interpretations. So this is the expression that we have in the paper, and it's composed by a bunch of terms that you have seen in the paper. So again, just to clarify here I was talking about the evidence, and here I'm talking about the log evidence, but it's the same thing, right? So so just to go very briefly over the various terms, this is the log evidence, and E, remember, is just a data set that you observe. This minus log P of theta hat is your essentially your maximum log likelihood. This theta hat indicates the maximum likelihood point under a specific model M. D here represents the number of parameters of your model. N is the number of samples. This J here is the Fisher information matrix, which is something that characterizes your model and is defined this way. We're not going to go over it in detail. And this is something else called the empirical information matrix, or observed information matrix, that again, we're not related to the Fisher information matrix, but we're not going to go into details here. And the idea is that this is an expression that is an approximation in the sense that you have this remainder here, small o of 1, in the sense that, you can write this log evidence on the left as a sum of these terms on the right, plus some term that will be suppressed when n gets large. So this small o of 1 contains things that get smaller and smaller as n gets larger. So as n gets larger and larger, this approximation here gets better and better. So some of you may recognize that the beginning of this expression looks something like the Bayesian information criterion that some of you may be familiar with. And in fact, this actually offers this is a generalization of the Bayesian information criterion. This is a well-known criterion in statistics to perform model selection. It's something that does, if I have to compare two models with different number of parameters, how much should I penalize the model that has more number of parameters because of its complexity? And this is how you penalize it. And you can see that in this expansion, we have that the first term, the one that depends on number of parameters, is the same as the one in the BIC. In fact, this is one way of deriving the BIC. But we also have other terms. And this is nice because it gives us these terms have geometrical interpretation. So again, to go over them, so what this is telling us is that when you're comparing different models, you have to look at, the likelihood is, how likely your data is under the maximum likelihood under your two models that you're comparing is obviously very important. So that's the first thing you look. Then you penalize, the more complex model, where complex, you penalize complexity as measured by the number of parameters. Then you penalize the more complex model where complexity is measured with the volume. So this thing here can be shown to represent somehow the volume of your model. So as in something like the number of distinguishable probability distributions that your model contains. So if you imagine, for instance, that your model is this kind of two-dimensional surface here, this abstract picture, and your observed data is this blue dot, and taking maximum likelihood is like doing this sort of projection operation here. So in this case, you would have this thing has 2 dimensions, so these 2, and then the volume is really the area of this thing, measure it in an appropriate way. that doesn't depend on choice of coordinates and things like that. And then you have another robustness term here, which is something that depends on how on the local geometry of your model around the point of maximum likelihood. Again, I'm not gonna go too much into details here, but this is just to give you an idea. So this is kind of generalizing the Bayesian information criterion by capturing further aspects, finer aspects of the model geometry. Let me check how time. Again, let me yeah. So now I was talking about, in the previous slide, I was talking about the fact that you have to choose a prior probability distribution over the model parameters. And here on the slide I wrote that I assume that this prior is uninformative. So what does it mean uninformative? So the idea is that, you know, just in the same way as we're establishing a prior, you know, between which model is true in case we have only two, you know, if you don't know anything, you might as well say, okay, they're 50-50, it's a uniform, right? And a similar idea, you know, holds when you're looking at your parameters and you don't know before observing the data, you don't know, you know, what values of the parameters are more likely than others. I mean, if you do have some ideas that comes from somewhere, by all means use it. But otherwise, if you don't know anything else, you might as well, oh yeah, I'm gonna just pick, you know, a uniform probability over the parameters. So that's kind of the spirit. The problem is that there's some like technical reasons why you cannot just say, oh, I'm going to pick a uniform probability over the parameters. So this is why we say this is an informative. It's kind of like a generalization of the concept. And this is what Jeffrey's prior is about. So the problem of picking a uniform probability over parameters is that it depends on how we define the parameters, right? You know, if I have a parameter that indicates a probability of coming up heads with a coin flip, right? You know, it's a P, it's probability of, say, heads. And I say, oh, I just want to take, you know, uniform probability over P between 0 and 1. But then, I mean, that's uniform, fine. But then if somehow I choose to, you know, parameterize my model as, you know, 1 over P, then all of a sudden this parameterization is not uniform. If I, you know, my distribution that is uniform over P is not uniform over 1 over P, right? There's some kind of internal inconsistency related to just saying, oh, I pick something uniform. So on one hand, you have to make sure that you're picking a non-informative prior in a way that does not depend on your particular choice of coordinates for the model. So Jeffrey's prior has this property. So it is invariant under changes of coordinates. So this is very nice. And another property that Jeffrey's prior has is that it assigns equal probability to volumes of the model that contain an equal number of distinguishable probability distributions. So this is the formal definition. It's something that's proportional to the square root of the determinant of, oh, sorry, I changed the notation here. This is Fisher information. It's the same thing that was J in the previous slide. This is just G, but it's the same. This is how it's defined. Don't care too much about now exactly how it's defined. I'm just going to give you a couple of examples to perhaps help with the intuition. So this is an example. So imagine, for instance, this is a very simplified example that doesn't quite tell the whole story, but you know, maybe can help some of you. Imagine that you have a model that looks like this, right? So you have that X and Y, you know, your data is a two-dimensional data, two-dimensional vectors. They're distributed as a Gaussian that is centered around some parametric point, mu of t, and with known and fixed isotropic variance sigma, okay? So this is mu of t is given by this expression here. It's a parabola, right? You can see I have represented it here as t goes from 0 to 1. Your center of the Gaussian kind of, you know, draws out this parabola here. And you can imagine that for each point of t, you have a little Gaussian kind of centered around that spot. So if you think of picking a, you know, if you want to parameterize this, if you want to put a prior probability distribution over this parameter t, and you want to say, oh, I just want to put a uniform probability on t, then you can imagine that you're going to select, say, just as, you know, if you imagine now drawing random values of t according to uniform probability distribution, right? So you're going to select points from the red segment here as often as you will select points from the yellow segment here, right? And that's fine, right? But the problem is that if you look at how, because the mapping between t and your model is nonlinear, right? if you look at how, like, if you look at like the size of the section of the model that is represented by this little segment, the red and the yellow, you will see that the yellow segment actually represents a bigger portion of the model. So that means that if you sample red as often as yellow, you will mean, you know, relatively oversampling the red part of the model, right? Because if you're choosing this yellow here as often as this red, but this one is smaller, it's like preferring the red part of the model. So this is just to give you an idea that uses a very simple geometrical intuition as to what does it mean to have, you know, problems because of the fact that, and this is due because of how you're like the fact that you're choosing a prior probability distribution that depends on the parameterization, right? Because it's uniform on T. Then if you parameterize this thing in a different way, then it's not going to be uniform anymore. And so just keeping straight, so in this case, for instance, the Jeffreys prior for over the parameter T would be something that looks like this. And for those of you who know a bit of differential geometry, this is basically the same thing as taking a uniform probability distribution over the arc length parameterization of this parabola. But if you look at the form of this this distribution, it's basically overweighting the end points, right? When T is far away from 1/2. So it's kind of counterbalancing this effect that I was telling you about, the fact that if you just take something uniform, it's going to prioritize the center of the model. Anyway, so this is just to give you an idea. But so this gives an idea of what it means to you know, for different parts of the model to contain different numbers of distinguishable probability distributions. But of course, probability models are more complicated than this very simple geometrical picture that we can visualize here. And this kind of leads me to another question, which was actually this is a very, very nice question. So regarding this topic, right? So why should all probability distributions count equally on the parameter manifold, given that they have different Shannon information entropy? The point is that it's not that all probability distributions count equally, only distinguishable probability distributions count equally. And if you think about it, you can define the entropy. So remember, model is a parametric family of probability distributions, right? Each of these things has an entropy, you know, assuming that we can define it. And so the entropy is a property of an individual member of this family. But distinguishability is a notion that relies on the relationship between a point on your manifold, so a particular probability distribution, and its neighbors, right? So in general, the relationship between Jeffreys prior and entropy is not straightforward. There are cases, though, in which, you know, simplified cases in which we can see a direct, more direct relation. I can give you an example here. So again, this all depends on what particular parameterizations we're choosing, but, you know, look at this example. So consider a binomial distribution, right? So this is the expression for the probability of k successes over n rows, where imagine you're flipping a coin n times with probability of coming up head theta. What's the probability of having k heads? Okay. So here I have plotted out at the top the shape that this probability distribution takes for multiple choices of your parameter theta. Actually, this is, instead of plotting probability of k as it's defined here, I plotted k over n, but it's the same, right? Just I fixed n equal 10 for this example. So you can see that, you know, when probability of success is very small, you tend to have a number of successes that are small compared to n, and vice versa. When you have something probability of success that is 1/2, you're going to get something that is nice and symmetric. Okay, so in this case, and notice that I'm parameterizing this model, this probability distribution family with this parameter theta. I could do something. I could choose another parameterization, but in this case, I'm choosing theta. So for this particular choice of parameterization, you can see that the models, when theta is very small. So if you look at the increments here I'm to go from one version to the other. You can imagine, your family is, you can imagine as being the sum total, I mean, the collection of all these variants of the probability distribution. Of course, there's a continuum of those. So here I represented a few of them, And the difference in theta between them is always about the same. It's always like 0.013, something like that. So you can see that as I change theta by 0.13, going from, say, the blue to the orange distribution here, there is a certain difference, right? But then if I apply the same change in theta to go from the purple to the pink, then the difference between probability distributions is much smaller. So in a way, you can see that the same change in theta affects a much smaller or bigger change in the shape of the probability distribution, depending on exactly where I am on the model manifold. And indeed, you can see that in this case, this is also, again, given this parameterization, this is related to the, so yeah, sorry, let me finish this. And you can see here below, I have plotted Jeffreys prior for this particular model in this parameterization. And you can see that it basically overweighs the models that are close to, gives more weight to the models that are close to theta, either 0 or 1, because as we can see, those models, there's more of them there. Basically, if you change theta in those regions, you have more distinguishable models. And you can also look at it this way. So the models that are either on the left or on the right, so theta very small or very large, they're also very narrow, right, in this plot. So they, you can think of them as having, a smaller, somehow they have a smaller entropy, right? Because they can only explain a few, they only encompass a few states, right? Whereas the models towards the center, they are broader. So in this case, that's one way in which you can think about the fact that neighboring models overlap more. But this is just, and in this particular case, it so happens that if you plot out, the blue line here is the entropy of the various distributions in the model, and you can see that it kind of goes nicely. It has a similar trend to what the Jeffreys prior is doing. But again, this is just to give you an example of a case where the two concepts kind of go together, but this is by no means necessary. Okay, so let me see. Do we have? Let me see. Also, Howard. Actually, let me start this presentation here. And okay, so Ron, what do you wanna do? Shall we move on to the answering a few questions about the retina paper?
Potentially, I know we're a little low on time.
Yeah, so we're a bit late, so yeah.
So if people have more questions about either grid cells or what you just presented, it might be easier to chat on that than maybe get the retina stuff in. But if people don't have a bunch of questions, then maybe we can try to do the retina stuff real quickly.
Yeah, maybe, okay, maybe what people think I can get started. So like I can, I can give some answers to some specific questions about the third paper. So there was one that was, I'm still sharing the screen, right? You still see my presentation. So for example, this one, right, has the asymmetrical distribution of on and off cells being found in other species than humans. So this one, if I understood it correctly, I mean, let's just get it out of the way because it's easy. So first of all, remember, so the data in the paper is not from humans, it's from guinea pigs. And also there's references, if you look through the reference list, talking about the fact that smaller receptive fields have been reported for off cells in guinea pig, rat, rabbit, monkey, and human. Now, the idea is that if you have smaller receptive fields, and if your off cells are building a mosaic of your field, then by necessity you need to have more of them. So this is actually an illustration of this idea. Now, I mean, this is like the two rows. Top row is on and bottom row is off. Now, if you want, you can squint and try to tell the difference between on and off, but that's not the point. The idea, these are two different types that in 2009 in macaque. But the idea is that to convey this idea of the retinal mosaic, the difference
Yes, I'm not sure if it's just me, Eugenio, but I think you're
exposed in
coming
such a way
in
that
and out a little bit.
oh, okay.
But mostly it came through that
Better
these
now?
are, we're talking about the mosaic and macaques.
Yeah. Yeah. So, you know, if receptive fields are smaller, you need more cells to tile the space. That's kind of the concept. And there is experimental evidence in other species too. So, yeah. So then let's see. Oh, there was a question about elements of the brain that are well conserved, particularly well conserved across species. So I think we came up with a couple of examples. Am I still choppy, or am I good now, Ron?
I think you're good now. Yeah. Or at least I'm not hearing any issues with the audio.
So we have. Let me show you this. So I wanted to give an example of, this is not, by no means an exhaustive answer, but I just had this example in mind. I think it's cool about, you know, aspects of the brain or neural circuits that are well conserved across evolution. So let me just give you this example. Let's see if I can actually put this thing at full screen somehow. How do I? Yeah. Oh no. Okay, sorry. I have to scroll through the whole thing. Okay, so this is an example from the cerebellum. So you know, the cerebellum, you can see it here in this picture, is involved with motor learning and motor control. Is very important area.
We've got another little fade out there, Eugenio.
And you have it. Ron, you wanna take over for a minute, and then let's see if my connection stabilizes. Yeah, if you want to switch to, yeah, I can stop sharing, so maybe I will get better.
Okay, I think it's stabilized for now, but I can go for a little bit. So I guess I've kind of already given my answer to the ones that's conserved earlier in my talk, where I was saying the hippocampus actually tends to be relatively well conserved, and the connections between the regions of the anteroral cortex and the hippocampus tend to be pretty well conserved amongst animals, which I find fascinating. Is very lucky in neuroscience, and brings up an important point of, like, the animal model that we choose to use is really critical based on the conclusions you want to make after that fact. So with rats and rodents, we can be more comfortable because there is this great overlap between species, versus, for example, I also work with an auditory lab, and the auditory experiments that are done in mice and rodents, the region is slightly different, the organization is different, the properties are different. One can imagine even how the animal is using that sense is quite different than how we're using it. So that you have to weight your conclusions from your data appropriately. I'm going to also keep looking at this next question. Okay, so I'll answer one grid cell question too from this. So how does it change in 3D? So that's a great question. So we don't actually have a lot of recordings from rodents in a 3D maze, because they're a little bit difficult for us to build, and at least before they were difficult to record from. So when we're actually recording neural activity, the animal has to physically have an implant on its head that allows us to get the electrical signal. And for up until recently, that signal had to be wired. We didn't have, like, now there's Bluetooth recording that you can do, where you can have a stage on the animal's head and actually have Bluetooth signals be sent. But because the animal is wired, trying to build a 3D environment and not have the animal either hurt itself or pull on its implant was rather difficult. So we don't have a lot of recordings from 3D. We did have, it was not published as a paper, but was presented as a poster, that if you put like a trench, kind of like a hole in the middle of not a hole necessarily, but like a little divot in the middle of a box field, it didn't seem to affect the grid pattern that much, which is quite fascinating. But this is an area of active research. Like, I know something VJ and I have been thinking about is the reverse, instead of having a trench, but having a hill, because at least the way that current models work, like having a hill in the box would change the amount of length that the animal is running over, of course, compared to its 2D projections. So we're interested to see how that might affect the grid pattern. But unfortunately, we don't have a lot of recordings from 3D mazes. You've been looking more stable, Eugenio, so I don't know if you want to give it another
Yeah,
Okay.
maybe I can try to finish what I was trying to say, and then we can pick a couple of questions, and then we can probably stop. Let me try sharing the screen again. I was trying to tell a story about certain. Yeah. Okay. So the idea, for instance, here that I like very much is that you know the circuit, the basic circuit that you know describes the cerebellum is relatively well conserved about across vertebrates. It's very nice and regular. But in particular, if we're thinking about if we look at the morphology of individual cell types, of course, this contains a certain number of cell types. But if we look at the morphology of this particular cell type, for instance, we have this big neuron here, which is called the Purkinje cell. And you know, it does more or less a general function across multiple animals, but its morphology you know, has some common traits. But you know, it changes a bit depending on specifically on what animal you're looking at. And you can think about it, you know, as a way of thinking, you know, evolution affects morphology, not just in, you know, at the level of the whole organisms, but also at the level of individual neurons. But the cool thing is that if you take another neuron of the same circuit, which is the cerebellar ganglion cells, by the way, this is the most numerous neuron in the brain, more than half of the neurons in your brain are cerebellar ganglion cells. So yeah, it's very important. The morphology of this particular neuron is extremely well conserved across animals. And you can see these images here. These are taken from different species, and some of these things are what we call living fossils, right? So the fact that this is so well conserved across species indicates that the morphology of this particular neuron has been very well conserved across hundreds of millions of years of evolution. So the idea is that this type of when this happens, I mean, this makes us think that you know this is this type of you know, when you bump into this type of facts, experimental facts, this always makes you think that this is a very good candidate for something that you know could be, I don't want to say explained, but you know well summarized and described by theories such as that look at the circuit from the point of view, okay, so what's the computational principle that's underlying this? What is the computational problem this is trying to solve? And is this particular element of the circuit to some extent optimized to perform this as well as it could? Because you know this hasn't changed so such a long time. So this is I think this is a striking example of what that question was trying to ask. Okay, so do we want to go back to, I don't know, do we want to pick like maybe one more question and then we can close?
I think that if that's allowed, that would be good.
Yeah. So let's see. So there's a couple of questions about the size of the receptive fields. There's somebody asking if they provide greater resolution, and another question about the fact that, oh, which one comes first? Is it a matter of size, you know, that you want more cells, or that you have smaller cells? I think, so I'm not sure. I think here it's hard to tell, you know, which one comes first, right? Because if you think about it in terms of evolutionary pressure, when you make these things smaller, you kind of get both things at the same time. And one important passage that was one important thing that, you know, that
We're getting a little dropout again, Eugenio, but
fields are smaller. Oh,
but I think you're okay now. I think it was just a quick blip.
Okay, yeah, so we're just saying that not only these receptive fields are smaller, but kind of the amount of computational resources devoted to each of them is the same as for the own cells. So you're really kind of increasing resolution in just maybe breaking this up into. So yeah, that's a nice question, and I don't think it has an easy answer.
No, it's I mean, I think I'll just briefly add to that something that really has been a theme in BJ's research, and that is something that plays a huge role, is that the generating an action potential, so actually one of these cells turning on firing a spike, is still the most metabolically expensive thing that the brain actually does. So like maintaining the correct electrical properties within a cell so that it can fire an action potential, that's what's taking most of the energy. So you have this real interesting balance between getting as much information as you can out of as few spikes as possible, so that efficient coding hypothesis. But how that also then lines up with the number of neurons is an interesting question. So it's like, would it be more advantageous to have more neurons but with a smaller receptor field so that it's firing on spikes overall, but then how much does it cost to the neuron? So it becomes a very complex optimization problem. But I will say, as you saw from at least 2 of these 3 papers, using this kind of efficient coding hypothesis does seem to get us some good insights, particularly on neuronal things that are in the periphery or more sensory, or have more like understandable receptor fields and receptor field properties. It seems, using this hypothesis, that how do we maximize information with the fewest number of spikes, that seems to be a guiding principle, or something that we at least can get useful information out when we take that approach to studying the brain.
Yeah, thanks, Ron. I think time is up more or less now, so we should, I think we can conclude. I'm gonna put up the slide again with our email addresses for another
that would be good.
moment. I guess you can save now. So if you have any other question, feel free to get in touch, and I think we will be able to share our presentations in case anybody's interested.
Yeah.
So yeah, thanks a lot, everybody, for joining.
Yeah, these are some great questions. It was nice to chat with everybody. Yeah, feel free to reach out to us if you have more follow-up questions. I know we didn't quite get to everything in the Q&A.
Okay, thanks, everybody.
See you guys.
Bye-bye.